سمحت لهم كلهم بالدخول مش عارف كده الناس دخلت.
Assalamu alaikum. Good morning, good afternoon, good evening for everybody everywhere. Thank you for every one of you for being here with us today. We are very pleased to be able to welcome you all to the new talk of the molecular genetics and biotechnology seminars. Before I get started, I would like to express my sincere appreciation to Dr. Ahmed Salam and his MGBS team for their great effort in organizing these talks. During the next 30 minutes, we will have a talk titled Short Exposure to Nitenpyram Pesticide that induced effects on reproduction, development, and metabolic gene expression profiles in the Drosophila melanogaster. This talk is going to be given by Professor Dr. Muhammad Ahmed Ibrahim. Dr. Muhammad Ahmed Ibrahim is a professor at Department of Plant Protection, Faculty of Agriculture, Asyut University. He was graduated and got his master's degree from the same faculty of Agriculture, Asyut University. Then he went to the States to get his PhD, which he received from University of California, Davis in 2011. And then after he got a postdoctoral fellowship twice from 2014 to 2015 and from 2019 to 2020. Dr. Muhammad is a member of international scientific societies, a lot of them, and he has like more than 65 research published in international journals. And he's also a supervisor for many master and PhD students. And he's also a reviewer in many international journals. Now we can have the talk by Dr. Muhammad. Please, you can start your talk for like 30 minutes. And after the 30 minutes, we will have like discussion for 10 to 15 minutes after. Thank you very much.
Okay, thank you very much. Thank you, Dr. Amir. Thank you, Dr. Abdel. First, I would like to thank Dr. Ahmed Atif Salam, the founder of the MGBS, and all the MGBS teams, of course. Today, I would like to give a lecture on titled Short Exposure to Nitenpyram Pesticide Induces Effects on Reproduction, Development, and Metabolic Gene Expression Profiles in Drosophila melanogaster, order Diptera family Drosophilidae. The contents actually will start with introduction, general hypothesis, materials and methods, results, and conclusion. Pesticides are the most effective tools in controlling pests. Actually, farmers consider it as the magical solutions for their pest problems, and believe it or not, without using pesticides, the global agricultural production will lose by 30%. But, of course, the frequently using of pesticides and the unwise and misuse of pesticides, it led to many issues like pest resistance, which is a big issue nowadays, and the harmful impact towards the total environment. Thus, it is very important to get and facilitate a new pesticide group or new strategy for control the insect pest. Neonicotinoids are the most, which is a group of pesticides. It's considered the most wonderful class of pesticide because its effectiveness, particularly on sucking pests like aphid, it has a unique mode of action in that it disrupts the insect nicotinic acetylcholine receptors of the nervous system. But of course, the overuse of pesticide related to this group emerges in agricultural contamination in all environmental aspects, especially in water, air, soil, non-target organisms like honeybees, which resulting in several impact issues in the whole ecosystem. So here in this figure, the left side, which is the normal side in the nervous system, when the acetylcholine and acetylcholinesterase break it down to move the signal of the what's happened in the after effect with the pesticides on the nervous system. And the second side, the B side, which is the right side, it's shown after treatment with the neonicotinoid pesticides, which shown like interrupting the sodium channel and moving of the sodium through the nervous system. And the result actually getting like paralyzed, the insect or the pest get paralyzed and the dead at the end. But what about the history of the pesticides? The history started in 1940 with the discovery of the pesticide DDT and using it against mosquito to control malaria. We know the malaria issue in the past and how many people actually got killed by malaria. But when we discovered the DDT like magical and reduce the killing people by killing the mosquito, which transfers the malaria. In 1960, and the appearing of the book called Silent Spring by Rachel Carson, published in September 1962. This book actually focused on the toxicological effects of DDT on the environment. And after the discovery of this book, the US EPA and the environmental movement start increasing the issue and about the impacts of DDT and pesticide in general. And this kind of movement and EPA start to say, oh, let's stop using DDT, let's start move forward and discover new pesticide which is more safe than DDT. In 1970, the discovery of pyrethroids group, which is another group of pesticides, and this group actually it's very effective and it has safe on all the environment, and mode of action is similar to hydrocarbon chlorinated pesticide like DDT. And for example, deltamethrin, which is an example of pyrethroid group, is 100 times powerful than DDT with the benefit of their safety. In 1990, the discovery of neonicotinoids group, which is our target of our lecture today. And in 2010, more than 65% of the scientists focused on the toxic effects of pesticides. So the aims of this study, first, to evaluate the toxicity of Nitenpyram. Nitenpyram is an example of neonicotinoid pesticides, and the aim here is to evaluate the toxicity on Drosophila melanogaster on third instar larvae and adults. The second aim is to examine the effects of sublethal concentration of Nitenpyram on detoxification enzymes, growth, development, and reproduction on Drosophila melanogaster. And the third one is to assess the changes in gene expression related to development and metabolism. Why we are choosing Drosophila melanogaster as a model? First, Drosophila melanogaster considered a standard model for toxicological studies of xenobiotics in insects. Second, the genome of Drosophila melanogaster has been completely sequenced. Third, the approximately 75% of the genes that are involved in the development of human diseases are evolutionarily conserved across the animal species. Fourth, the structural and functional genome has been found to be comparable to human genomes. Last one, Drosophila melanogaster has short life cycle, easy to maintain, of course, in culture in the lab, produced many eggs, and has low number of chromosomes, only 4 pairs, which is make it like excellent for a standard model for our study. So the life cycle of Drosophila melanogaster is divided into 4 stages: the embryo, larvae, pupae, and adults. The time length of the stages is approximate and is shown in hours for embryos and days for larvae and pupa. The general hypothesis that we are developed here, and we want to answer it as like a yes or no, it depends on our results. The hypothesis here is the short exposure to Nitenpyram and at low concentration sufficient to induce significant physiological disorders in animals, predictive and negative impacts on the health of the studied animal model. So this is the hypothesis, and we need to answer it. So materials and methods, the insect or the basis that we are used here in this study is Nitenpyram with purity of 99.9%. We actually purchased it from Sigma Aldrich from United States. The type of the Drosophila that we are used in this study is called wild type W1118. And this type actually we obtained from Dr. Joanna Shu from the University of California, Davis. The Drosophila preserved at 25 degrees and relative humidity of 70% and reared in 50 milli vials containing potato-based medium with what are purchased from Carolina company from the United States. Cystiasis, which we are conducted on the third instar larvae and adults as well by using 5 different concentrations mixed with the food and 3 replicate for each concentration, and the mortality percent was recorded after 24 hours of exposure. Development effect assays was done by applying the sublethal concentrations 1/10 of the LC50 values, which is in this experiment 0.71 microgram per milliliter. All development effects parameters such as larval and pupal duration, adult eclosion duration. Eclosion here means the emergence of adults from their pupal case. So the meaning of eclosion, because we will repeat it again and again, so I would like to emphasize here the meaning of eclosion, which is the emergence of adults from their pupal case. And plus number of eggs and lifespan, which is a type of developmental effect parameters we studied in this experiment. And of course, the lifespan here is the period of time between the birth and death of an organism. The RNA extraction, of course, we did the RNA extraction and quantitative real-time and reverse transcriptase PCR to measure the level of gene expression for certain genes. I will talk about it later. So the RNA extraction, we did it by homogenizing the Drosophila samples after treated by 2 different sublethal doses, the 1/3 and 1/10 from the LC50 value. After the homogenized the Drosophila samples, we isolated the RNA using the RNA isolation kit from Zymo Research from California. Then the RNA was converted to complementary cDNA using applied biosystem, high capacity, and we're using reverse transcription PCR. The gene primers were designed using primers 3 design software. Then gene expression were detected using the real-time PCR. In this experiment, we used 10 genes and we analyzed them in this experiment. And these genes actually is related to detoxification enzymes, developmental and behavior, and metabolic, of course, parameters. The first 3, the CYP12D1 and CYP9F2, CYP4AE1, all these 3 actually is detoxification enzymes, the mixed function oxidation enzymes. The enzyme is called B450. The second one is GST-D2, is another detoxification enzyme. It's called glutathione S transferase or GST. RYR is the hemocyte proliferation. IM4 is immune response. ETG7 is lifespan. PLE is a male mating behavior. DDC is a female fertility. The SXE2 is a lipid metabolism. So all these genes we have been chosen because we need to know the gene expression, all these gene expressions after the treatment by the sublethal dose, two actually sublethal doses, 1/3 and 1/10 sublethal dose of the LC50 values. So statistical analysis. So first, actually, we corrected the mortality percent using what's called Abbott's formula, and the tolerance ratio calculated by LC50 value for adults divided by the LC50 value for the third nestal larvae after 24 hours of exposure. The developmental effects of QRT PCR is data were determined by student t-test. And of course, the figures we created using the graph-prism software. The results, the table number 1, it's showing like Drosophila stage, the larvae and adults. And the LC50 value of larvae was 7.12 microgram per milliliter, and for adults was 38.07 microgram per milliliter. However, the tolerance ratio was 5.35-fold. The developmental stage effects showed that eclosion duration, which is the last column on the right side, was significantly delayed compared to control. The Drosophila that treated by Nitenpyram was almost 11 days, whereas the control was 6 days. So if we are comparing the eclosion duration compared to the control after treatment, so the Drosophila that treatment by Nitenpyram was almost 11 days. However, the control one without any treatment, it's like 6 days. So it's like a big delay. The lifespan was shortening after treatment by Nitenpyram. However, the lifespan of treated with Nitenpyram was almost 40 days, while the control was 50 days. Pupation rate percent was shortening too after treatment by Nitenpyram, which was 60% compared to the control, which was 98%. Eclosion rate percent. The eclosion rate percent was shortening too after treated by Nitenpyram, which was 40% compared to the control, which is almost 80%. Even number of eggs for each couple of Drosophila was shortening after treated by Nitenpyram, which was 20 eggs for each couple compared to the control, which was 50 eggs. So that's the developmental assays. So now about the gene expression levels. As we mentioned before, we analysis like 10 genes, and the genes actually expression results presented in these figures. The level of selected cytochrome B450 genes expressed in Drosophila melanogaster. The CYP12D1 was stimulated by both sublethal concentration. As I mentioned before, we used two sublethal concentrations, the 1/3 and 1/10, and the CYP or the CYP12D1 was stimulated, and the gene expression, the maximum gene expression induced by about 9.3 and 11.9-fold respectively. That's figure A. So figure B, the CYP9F2 was upregulated by 7.01 and 9.3-fold for both sublethal concentrations. This is figure B. Last one, the figure C, which is CYP4AE1, was increased by 4.11 and 7.5-fold for tested sublethal concentrations. This is the C figure, actually. So all the CYP or the detoxification enzyme B450 was gotten upregulated. The gene expression was upregulated. On the other side, the genes STD2 was not significantly regulated by both sublethal concentration, and that's shown actually in figure D. Figure E, which shows the gene expression, the RYR gene expression, and RYR is actually hemocyte proliferation in Drosophila, was upregulated by Nitenpyram, especially at higher sublethal concentration, almost 5.9-fold. That's figure E. IM4 gene, which is associated with the immunity system, was found to be significantly upregulated after treatment with both sublethal concentration of Nitenpyram, and it's like 6.1 and 8.4-fold respectively, compared to the control one. That's figure number F. G, the ATG7, which is a vital factor in the adjustment of Drosophila melanogaster lifespan, was found to be downregulated by Nitenpyram by 5.3 and 16.5-fold respectively. That's graph number G. The second one in this slide, the BLE, which is H. This gene actually regulated mating behavior in males, was significantly downregulated as well by the pesticides at both sublethal concentration by 9.1 and 14.7-fold respectively. And I, the last figure, the right one, GDC, which is associated with the female fertility and eclosion in Drosophila, was downregulated to, especially at 1/3 sublethal concentration of Nitenpyram by 8.3-fold. The last one in the figure of the gene expression level, the SXE2, the G1, the one in front of us, which is regulated the lipid metabolism. And this gene actually was suppressed by Nitenpyram, particularly at the higher sublethal concentration, and the suppressed actually by 14.3-fold. That's all the effects on the gene expression, the 10 gene expression that we have been choosing in this. So in the conclusion here, we would like to emphasize on that Nitenpyram is more potent on larvae than adults of Drosophila melanogaster. Nitenpyram prolongs the developmental time for both pupation and eclosion. The oldest genes, the SYP12, the SYP9F2, the SYP4AE1, RYR, and IM4 genes were significantly upregulated. These genes, ATG7, PLE, DDC, and SXE2 genes were significantly down-regulated. To be more given more explanation, and actually our justification for what's happened is that. So what's happened actually is when the Drosophila treated by the sublethal concentration of the Nitenpyram, they are getting stress from the xenobiotics, which is the pesticide in this case, which led to increase the CYP, which is the B450 or the mixed function oxidase enzymes genes to detoxificate the toxic. So when the pesticide getting inside the insect or treated by the insect, the genes actually got to be increased to detoxificate the pesticide, to get rid of the pesticides. Then get the increase of the immune system. The immune system got an increase too, and the hemocyte proliferation in the blood of the insect, which led to, because that's happened very quick and happened suddenly, that's led to disrupting and decrease in the gene expression of all the developmental gene expression parameters, such as the PLE, which is male mating behavior, the DDC, associated with the female fertility, the ATG7, which is associated with lifespan, and the SXE2, which is the lipid metabolism. And all these genes actually, especially the lipid metabolism, the SXE2, is actually getting down because the body need more energy and breaking down the lipids to face the toxic of the pesticides. And of course, we use two sublethal doses to get the more understand of what will happen if we treated the Drosophila melanogaster as a model here by Nitenpyram. So this study actually published in a journal called Science of the Total Environment in 2022, and the journal actually is Q1, impact factor 7.963, and site score 10.5, and the journal is 25 or 25th out of the 274 in the environmental science by Clarivate. We got actually a certificate from Elsevier and from the journal about our publication. Finally, I would like to thank Dr. Chris Vogel, who was my supervisor in the United States of America, University of California, Davis, plus Dr. Malafia from Brazil, who actually did like a collaboration with us to help us actually in certain points and certain explanations, which is really useful and important for us. Of course, I would like to thank everyone, all the audience, all the MGBS teams, and of course, I would like to thank Dr. Ahmed Asif Salam, the founder of the MGBS, and all the audience. And yeah, thank you very much. Hello, Dr. Abdul, Dr. Amir. Hello. Hello.
Hello? Hello? Do you hear me, Doctor?
Yeah, yeah. Yeah, I hear you, Dr. Adnan.
Sorry, it's Amir now. Sorry, my
yeah,
mic was
sorry.
muted, so I couldn't. Thank you. Thank you very much for this very interesting talk. And now we have like 10 minutes for discussion. If you have any one of the audience can write his question on the chat room, and I can read it for Dr. Muhammad. For starting, I can ask you a small question here. So you studied the effect of this pesticide on only the Drosophila melanogaster. What do you expect if the effect will be on the plants itself? So do you try to investigate this or not?
Actually, as you know, we are focused on the effects of, in general, we are focused on the effects of insecticide or pesticides on insect pests. But of course, there is an effect on the plants too. But until now, we don't actually do any conducting any experiments. But it could be a good point in the future. But right now, we only focus on the Drosophila as a standard model. But in the future we can focus on the plants, and it could be a good point.
And do you expect that you will have the same effect of on the insects, on different type of insects other than Drosophila melanogaster, or they will change?
Actually, I can't tell yes or no until I do the research, and after that I can answer this question. But as you know, Drosophila is like a model, and it's an insect. So my prediction actually, it could be like for somehow there's bad effects on the same parameters like behavioral and metabolic developmental parameters of another insect. But to be sure, I can't tell you exactly what will happen until I'm doing the experiments. So the question is like, give me like a time to it, so I can tell you.
Okay, thank you very much. Do we have more questions? If you want to ask yourself, you can raise your hand and I can unmute your mic if you want. No more questions. Can wait like a couple of minutes now. Amal want to ask a question, so I will allow mic for her. Now we can talk. I'm trying to allow her mic. No way. So can you write your question and I can read it for her?
Yeah, she can write in chat room the question.
Yes.
So you can answer
it. I think she should unmute herself.
Okay.
I tried, but I couldn't do that.
Yeah,
Okay, yeah, she can write the question in the chat room, so it will be much easier for her.
Do we have other questions? So I want to ask all the audience to register at the link which is in the chat room, so they can get the certificates for attending these seminars. And I also want to announce that this lecture are recorded and it will be published on the website of the MGBS. Now I think I'm waiting for Amal for her question, but she's not ready yet. Shall we wait more?
Dr.
Okay,
Amir,
so the question is like, may I ask you, sir, have you worked to getting rid of the insects? That's a really good question. To be honest with you, we will not able to get rid of the insects because for somehow insect is really has like a benefits in the whole environment and ecosystem, because each insect there is another microorganisms or organisms that depending on it or on them. So we can't get rid of them completely, but what we are doing, we got reduce the number of the bad insects, and be careful about what's called bad insect, which is called the pests. So what we are doing all the time in plant protection, we get reduce the number. So the insect or the population of the pests didn't affect my economic issue actually. So we are not able to get rid of insects, but what we are doing again, we are reduce the number of the bad insects to be in the safe side. Thank you, Amal.
We have another question here. What are the methods used to measure the effect of these materials on insects? Do you measure the weight of the insect or how?
That's a really good question too. So in certain cases, we actually calculate or count the dead and the live one, the live and dead insects based on the treatment of our pesticides. That's like acute bioassay. And another one, we get like doing like measuring like the lifespan. So as we mentioned in the life cycle, we have employ larvae, pupa, and adults. So in the developmental parameters, we are keep in touch with all this kind of stages after the treatment of the sublethal concentration of Nitenpyram, the pesticides, and we figure out if there is like abnormal malformation or the dead one, or actually the sex ratio, the male and the female percent. All these parameters we are calculated and we put it in the paper. And of course, like the gene expression was this the most effective way to get the effect of the pesticides, the Nitenpyram, on the Drosophila, on larvae and adult stage. Thank you, Muhammad.
So Amal again is asking, is there any safe, eco-friendly method for that? I don't know what she's meaning, but maybe you have an answer.
Is there any safe, eco-friendly method for that? What does it mean?
I think sure, like getting rid of insects. I don't know.
So now we are what is called alternative, which means pesticide alternatives. We are using now a safe pesticides, what's called green chemistry or green pesticides. Also, we use different methods like pheromones, like insect growth regulators. We are using the sterilizing insects. We are using different methods to be sure that these methods will be safe on the environment and ecosystem. Plus, if we are need to use the pesticide, we are using the pesticides that the Ministry of Agriculture says that's okay, because all the world actually using pesticide, all the whole world, but they are using the pesticide which is green zone, which is the safe, using the effective application way. That's why we don't have any. I mean, like Canada, America, the developing countries actually using it, and we don't hear any problems actually. compared to us. So we start using this pesticide alternatives methods, and and biological control like predators, parasitoids, microorganisms. There is what's called biopesticides, which is the active ingredient there is not a chemical, it's like microorganisms. So that's more specific, and it has less effect, less bad effect on the environment. Also, we use like a plant extract, which is another strategy. We are using it instead of using the pesticides all the time. So that's all. This method is called eco-friendly and like a save on the ecosystems. Thank you, Amal, as well.
She wants your email?
Yeah, I will write it right now, so it's not. So yeah, here is my email address.
Thank you very much, Dr. Muhammad. We are very happy that we heard from you, and we want to hear from you more and more, maybe in the next seminars. Thank you for all the audience and the attendees. And now I can finish the seminar. Thank you very much again, and see you again. Bye. Goodbye.